Okay, it looks like it's top of the hour. Thank you everyone for joining us today for our webinar. Just a couple things before we get started. During the webinar, you're more than welcome to send us questions via the chat function throughout the webinar, and we'll try to get to those at the end. And your certificates for the U.S. attendees will be available on Monday by logging into your account and downloading those there. Irene, if you'd like to introduce our speaker, go right ahead.
Thank you, Kim, and thank you all for joining us today. I'm Irene Zoppi, the Senior Clinical Manager for Education in the United States. Last year, during the midst of the coronavirus pandemic, Medela convened a global roundtable discussion with leading human milk scientists and researchers about the SARS-CoV-2 virus. A summary of the roundtable discussion was just recently published in the February issue of Frontiers in Pediatrics, and it's available as a free downloadable article via PubMed. A white paper describing the impact of COVID-19 on maternity clinical practices and breastfeeding support, along with a call to action, was also developed as a result of the global roundtable and is available by visiting medela.com. And Kim sent a link to the white paper today with today's link for the webinar of Dr. Powell. So today's webinar by Dr. Rebecca Powell is the second in a series of follow-up leadership presentations designed to continue to raise awareness of the coronavirus on breast milk initiation. And that series is entitled Breast Milk Initiation in Crisis, the After Effects of COVID-19. Dr. Powell will share her current research on the human milk antibody response to SARS-CoV-2 infection and recent data on the milk antibody response following vaccination against COVID-19. Dr. Powell, we're ready for you, so go right ahead.
Great. Hi, everyone. Thank you so much for the invite. So one second. There we go. So today, I'm going to talk to you more about our research on the antibody response against SARS-CoV-2, or more commonly known as COVID-19, in human milk. I'm in the Division of Infectious Diseases at Mount Sinai School of Medicine in New York City. So this webinar is sponsored by Medela. They did not have input to this content, and I don't have any other conflicts to disclose. Today, there'll be a few objectives, and just to touch on them here. So firstly, for everyone who's less familiar, one objective is to learn just about the basic immunological components of human milk, to understand the antibody response in human milk and how this is different from what we call the systemic response, which is what you would see in blood. And then to learn about the potency and durability and function of the antibody response against COVID-19 in human milk. And then at the end, I'll give you a little peek into what we're finding about the immune response to vaccination, and we can compare that back to what I'll have talked about here of the response to infection. So just some background. So antibodies, we've all heard about them. An antibody is a protective protein that's produced by the immune system in response to the presence of a foreign substance. So our bodies can recognize a foreign invader, and they can also recognize self proteins, our own proteins, and the immune system, part of its job is distinguishing between the two. So you can see here, this is a cartoon diagram of an antibody, and just to keep in mind, there's two ends to the antibody. So it's got this Y shape when we diagram it like this. Of course, this isn't really what it looks like, but this is the general gist of it. So one side is what we call the variable side with the V's here. And you can see that would be what is different for every kind of antibody in us. So there's the antibodies that are going to be very specific for the proteins made by the SARS-CoV-2 virus. And then there's going to be antibodies specific for flu or for other foreign substances. And then you can see here what has the C's on it. That's what's known as the constant region of the antibody. That does not differ among all the types of antibodies in our bodies. And that side of the antibody, I'm not going to really get into today, but it does help perform other functions of the immune system related to the antibodies. So as I said, antibodies recognize, and they latch onto, they really stick to, in kind of a non-permanent way, but still very strongly, they latch onto the foreign substance in order to remove it from the body. And so when an alien substance enters the body, the immune system is able to recognize it as foreign because its proteins are very different looking to the immune system from those found in our own bodies. Now, it's not that simple. So there are many different types of antibodies, and they're not all created equal. I'm not going to get into all of these today, but just so you're aware, there's many different classes of antibodies, and I will touch on some of them in what I described today. So you've probably heard of IgG. So IgG is what's most dominant in the blood, and there's different subclasses of IgG that perform somewhat subtle different functions, and I won't get into that today. There's IgM. Some of the COVID-19 tests, they detect IgM and IgG. IgM is produced really early in the immune response, and it also tends to look more like this star shape. So it's actually multiple antibodies connected. And then IgA is very important for what I'll describe today because IgA is what's dominant in milk. It's also found in blood, but it looks different and it has different forms. So in blood, it tends to be this monomer. That's what we call just a single molecule. And then in our mucosal secretions, such as milk, but also in our respiratory tract, our digestive tract, and other secretions of our body, you would actually find more of what's called a dimer, which is two molecules connected, or even higher order molecules as well. And then there's a couple other classes that we won't talk about today. So as I said, 90% of milk antibody, roughly speaking, is IgA. So it's quite important to the discussion today. So this 90% IgA is then along with about 8% or so of IgM. So this is the total composition. This is not specific to any type of antibody, you know, against COVID-19 or flu or what have you. This is the total composition, and it's an estimate. There's also a lot of variation from person to person, and there's been different studies that, you know, they're not always the exact same numbers. It's also geographically specific. So in different parts of the world, people who originate from different places may have a slightly different composition to what types of antibodies are in their milk. But it's about 90% IgA and about 8% IgM. And those antibodies are nearly all in what's called secretory form, which is very important to their function in milk and in other secretions. So a secretory antibody is polymeric, so meaning it's made up of different kinds of proteins. And these, in this case for IgA, and most of the time it'll be a dimer, so it'll be two IgA molecules connected by another protein called J chain, and then wrapped up in another protein called secretory component. And then I have a little cartoon here to diagram that. So again, in the blood, we do find monomeric IgA. There are two different types, but we won't get into that. And then in the secretions, we tend to mostly find dimeric IgA, again, connected by another protein made by B cells. B cells are the cells that make antibodies. The B cell makes another protein called J chain, and that connects these monomers to each other, forming the dimer, right, the duo. And then finally, this complex is wrapped up in secretory component, which is very important to its function. So there is some IgG in milk, very minimum amount, about 2% of total milk antibody is IgG. And that mostly comes in passively from the blood, from the serum of the blood. And there may be some local production from certain localized immune responses near the mammary glands, in the lymph nodes, and then some of that also will come into the milk. So it may be a little bit different from what you would find in the blood system, but most of it's coming in from the blood. And the B cells that actually make the secretory antibodies, so mostly IgA, secretory IgA, and some secretory IgM, those B cells actually come from other mucosal tissues, but mostly the gut. So they mostly come from what's called the GALT, the gut-associated lymphatic tissue. And there's a little bit of respiratory mucosal involvement as well, but predominantly those cells are coming from our gut, our digestive tract, our small intestine specifically. So this is known as the enteromammary link. And so if you think about this evolutionarily, it makes perfect sense, as evolution tends to do, where the mother, all the pathogens that come into her environment that she's really ingesting for the most part, swallowing or inhaling, those will end up down in her gut, and then there'll be a immune response in the gut, specifically in the small intestine, in the specialized tissue of the immune system called the gall. And then that will generate an immune response that's responsive to what's in her environment, especially to gastrointestinal pathogens, which are so dangerous for very young babies. And then those cells will transit, they will travel to the mammary gland. They're programmed to do that. And then the mammary gland will then produce antibodies into the milk that match what's in the mother's environment. So then the baby is then protected in their gastrointestinal tract and their respiratory tract from those pathogens that really could kill them, right? So it's really perfect. And it makes a lot of sense. And there's a lot of evidence. There's some studies in mice that demonstrate that this is the case, and in some other mammals. And so some of this evidence, I won't get too complicated, but the B cells that are in the mammary gland, we know that the proteins on the surfaces of those cells actually are almost identical to the ones that you find in the gut. So that's one piece of evidence. And that's very different from B cells you find in different parts of the body. The milk secretory IgA actually has a lot of specificity for the pathogens of the digestive tract compared to if you looked in the blood or in other areas of the body. And then an interesting piece of evidence just from one mouse study that I'll touch on is that there's a strain of mouse that have really long intestinal tracts, really long small intestines, and those mice actually have increased B cells in the mammary gland and increased secretory IgA in their milk compared to regular mice. So this is some of the science that leads to us concluding that there is indeed this enteromammary link. So now I'm going to talk a little about the functions of antibodies. And there's many different functions, and I won't really get into most of them today, but let's just go over it a little bit. So as I said, there's two parts to an antibody, generally speaking. There's the variable side, which is very specific for the pathogen in question. And then there's the constant side that is not specific to the pathogen, and that is just going to do its function that it's going to do no matter what the context. So in the variable region is also known as the FAB fragment. For old scientific reasons I won't get into, but the FAB or Fab mediated function, so meaning having to do specifically with that variable region that is specific for, let's say, COVID-19, that function is really to block, it binds to the pathogen and it blocks the pathogen from infecting a cell. So that's called neutralization or a blocking of infection, and that's really critical. Then there are other functions that are actually mediated by that constant region of the antibody, just so you know that there's many functions of antibodies, but again, I won't really get into those today. So now let's get a little deeper into SARS-CoV-2. So, you know, we all know probably way too much about COVID-19, way too much about SARS-CoV-2 at this point. You've probably heard of the spikes, the spike protein on the virus. So the SARS-CoV-2 spike is the protein that's responsible for allowing the virus to attach to and fuse with and really infect the membrane, so the coating of the host cell. So without the spike protein, the virus really couldn't infect cells and it would be harmless. So the spike is what allows it to infect the cells. So it's a very important target for antibody to bind to block that process from happening. And that's really what is targeted by the vaccines that have been designed. That's what really the most potent antibodies that we make after infection are going to be against what's the most visible to the immune system, which is what's on the surface. That's called the spike. So there's a cartoon diagram of that down here. So this is your SARS-CoV-2 with its spike proteins. And of course it has other proteins that make up this virus. And then we see the spike protein, it looks like a flower here. It's actually 3 spikes that glue together to something called a trimer, meaning there's 3 of them. There's different regions of it, and one important region is the receptor binding domain. So that's specifically the spot that the receptor on the cell that is what matches this puzzle here of the viral spike, what it's going to bind to. So the spike has a very strong affinity. It recognizes this protein on our cells, which is what makes it so dangerous, and that's what it'll bind to infect our cells. So if we didn't have this receptor called ACE2, it could not infect our cells. And unfortunately, this ACE2 is an enzyme and has its own function, but it's attached to the cells of many different types of tissues in our body. So lungs, arteries, heart, kidney, intestines. And this is why the virus can be so dangerous and why its symptoms can vary so widely, because that receptor is actually on many different tissues in our body, and not everybody's the same in how they express that protein on different parts of their body or how much of it they're going to express. So that's why there's a lot of variable symptomology after infection. So obviously spike, it's very important, and it's the main target for what's called a functional antibody response, functional antibody binding. So now we'll talk a bit about COVID-19 in babies and young children. So we know, it's pretty common knowledge now, that COVID-19 pathology, how it affects people in children, young children, infants, even, older children, it's really typically mild compared to adults, thankfully. But still, about 10% of babies under 1 who get infected with SARS-CoV-2 will experience severe illness that requires advanced care. So they will very likely recover in almost every case, but they might require really advanced care and hospitalization. And globally then, of course, there are a lot of infants and young children who have been made quite ill by this virus, even though, on a case-by-case basis, it does affect children much less than adults. And as well, again, globally, young children really are still responsible for a lot of the spread of this virus on the whole. And also, I'm sure you've heard of this MIS-C, this multi-system inflammatory syndrome in children that's associated with COVID-19. So this is thought to be an autoimmune response. Its symptoms do vary widely. It can be very dangerous. It can be fatal. It's rare, but again, in a pandemic context, you can definitely see it, the cases occurring in hospitals of children, infants and children who were exposed to the virus. They didn't even necessarily know, and then they can end up a few weeks or months later actually quite sick from this autoimmune response. So there's many reasons why protecting this population is really essential in the pandemic. And one protective mechanism might be the passive immunity from breastfeeding by a previously infected mother or milk donor. So the milk immune response to SARS-CoV-2, before really I started my work, which was early on in the pandemic here in New York City, and there's other groups who also have some similar studies going on as well. Before that, nothing was known, of course, about the milk immune response to SARS-CoV-2 because it was such a novel virus. And there was a lot of concern that the virus could be passed in milk, which since has essentially been demonstrated to not be the case. So it's recommended that that infected mothers do continue to breastfeed, that they establish breastfeeding if that's what is desired, and that there's pretty much no risk directly from the milk for the infant to contract SARS-CoV-2, although of course they're still in that environment of an infected parent. So masking and other hygiene methods are necessary, like good hand washing and so forth. So, understanding the human milk response to infection and vaccination is really essential because there's been so much misinformation, at every level, and everybody needs to know and understand what's going on. And, it's sometimes very difficult to get the right information. So, parents and lactation professionals, healthcare professionals, and scientists, everybody needs this information. It's really essential. And we need to use science to inform public health rather than what was especially going on early on in the pandemic of so much hearsay and so much misinformation about breastfeeding and COVID-19. And then, not only is this, of course, critical for public health and for infant feeding and protection of this population, but an extension of this that my lab is studying is that if SARS-CoV-2 milk antibodies are produced at a high amount, which is called high titer, and they're good quality after infection, then these antibodies, secretory antibodies, they're actually really durable. So they're very different from the IgG that's mainly found in serum. They have that secretory component that I mentioned that's wrapped up in this secretory component, which protects it in the infant mouth and gut, in the milk, from those relatively harsh environments. So secretory IgA is really special and really could be an innovative therapeutic for COVID-19 for anybody. So we could extract that antibody if we see that it's coming, in high amounts in milk after infection. We can purify it from milk. It could be used therapeutically for anybody. As a really directed treatment in the respiratory tract, or maybe an oral treatment as well, because that's where those antibodies are designed to be. So that's something our lab is studying as well. So for our study, which really was initiated in early April in 2020 in New York City, and then broadened to a national study, we've now recruited just over about 800 lactating participants who have recovered from COVID-19. And so our recruitment really had an immense response, a wonderful positive response when we started recruiting because of the time we were in, and it really caught on. So, I use social media for my recruitment, and, it really was so successful, which was great. So you may have seen, in the news, this recruitment really caught on. And so there was a few articles, and that only made the recruitment go better and better and better. So we got so many people in this study, which is really excellent and what we needed. And you can see, we just kept getting that word of mouth going. So we've had a great recruitment. And so in our study, to look at the antibody response to COVID-19, what we do is we get about 30 mils, which is about 1 ounce. So we don't ask for much. We get about an ounce of milk from study participants. So these are people who've recovered from COVID-19. They pump it home, so which is especially important, for a low contact or no contact situation, especially, in the height of the pandemic. People did not want to have to go anywhere, or they could not go anywhere. So they pump it home, they freeze the milk samples at home, and they wait for us to pick them up. So all the participants I'm going to show you today had a confirmed past SARS-CoV-2 infection. And the milk was obtained about 4 to 6 weeks after infection. And in some cases, they were able to give longitudinal samples, and many of these people are continuing to save milk for me now. And so they're saving monthly samples after their initial sample, again, in their own freezers. And then we schedule pickups, and we have milk shipped in, every so often from these participants. And the goal is to really have people in the study for maybe as much as 2 years, which is, not everybody, many people don't breastfeed for that long, but fortunately some people do. So we have a big recruitment because we know a lot of people will naturally wean and drop out of the study. And we want to know what's in the milk in 2 years, right? That's something really important that has absolutely not been examined for any infection, to understand what we would see so much later, which of course has many important repercussions for long-term breastfeeding, for future babies, etc. So again, I picked up a lot of these early samples. Some of my staff have picked up other samples. We have couriers, and then also we get milk shipped in by Milk Stork, which is, again, it's paid for by some gifted support from Medela. And we have all this milk coming into our lab. And then as well, we also have, I'll mention, control samples, which were obtained before the pandemic. So we know that that milk could not come from people who are exposed to SARS-CoV-2. And those kinds of control samples are really important to set up in our analyses what we consider to be positive or negative for specificity to COVID-19. And I'm going to just touch on how we actually examine the milk for specific antibodies. So we use a method called an ELISA, which maybe you've heard of, depending on your background. So an ELISA, it's a plate-based assay, meaning it's done on a solid surface using molecular grade plastics in really a rectangular plate with a bunch of different sample areas called wells. And it's a technique designed for detecting and quantifying soluble substances, meaning they can be dissolved in a liquid, such as the level of antibodies in milk for a specific specific for a viral protein, in this case, spike-specific antibodies in milk, right? So the way it works, I mean, it's very, I did not invent this. This is an age-old, very tried and tested method, but we have made it work really well for milk. So what we have is we have the spike protein that's been synthesized. We don't cut it off the virus. It's been synthesized, what's called recombinant protein. So it's created in a lab based off of the known genetic sequence for spike. We can do that. And then that is really just glued down to this plastic surface because the plastic's coated with a substance that makes proteins stick to it. So that's immobilized on the plate. And then that sits overnight, we clean it off, and then it has the spike stuck to the plastic well. We then add on the milk. The milk is added, it's spun, so the fat's removed, the cells in the milk are removed. So we clean it up. So it's really like skim milk that we use, and it's not diluted with anything in the initial screen. And actually that works really well in this ELISA. And that's very different from blood plasma, because plasma you have to dilute to actually be able to do this with. With milk, it's great. You can just use it straight away. And then we do dilute the milk to get a sense of the strength of the response, and I'll show you that in a few minutes. So you add the milk, then you let it sit, you wash off what's not stuck down to the spike protein, then we add on what's called a secondary antibody, which is, that antibody is made by a company and it can recognize human antibodies. So that's pretty cool. And you can just buy that and it has a fluorescent tag on it. So I don't need to do that. That's made by a commercial company. And then you wash it off, and then we have a machine that can read this fluorescent tag. It can detect it. So you can't see by eye what's happening, but it's fluorescent and the machine can detect it. And then we basically quantitate the amount of light, which you'll see listed as the optical density, or the OD. So that's just the amount of light that comes off of this plate, and that's equivalent to how much antibody was there in the milk. So now I'm going to try and go slowly through these results so that everybody can understand, and it may feel a little bit dense, but I'm going to try and go step by step. So here's what we found. As I said, initially we used the undiluted skim milk. About 4 to 6 weeks after infection is when we got these samples, and in this first screen of the samples I'm going to show you, this is from 75 COVID-19 recovered donors, as well as 20 milk samples that are controls. And this was screened looking for IgA, which again is the most dominant class in milk, IgA that was specific for our spike protein from SARS-CoV-2. And so what we found in this initial screen, again, you can quite plainly see that most of the samples from COVID-19 recovered donors have a much higher OD value, meaning amount of antibody that's specific for SARS-CoV-2 compared to the controls. So mathematically, we can use these values from the controls to determine a positive cutoff. So anything above this line, we are considering positive. Anything below the line is what you would consider just background levels of light. It's negative. Okay, so 88% of these samples were positive for spike-specific IgA. And again, this is undiluted milk. So this is what the baby's drinking, right? So that's really, most of them. And obviously there's variation. There's always human variation, and we don't really understand that. There is a variety of levels of antibody here, or maybe different strengths of the antibody. We don't know, but they're all positive, all above this line. And then following this initial screening, we did not fully analyze every single one of those, because that's really a lot of samples, and this is still kind of in our preliminary phases of research, and we're going to do a lot more in-depth investigation of many different time points from all of these people, looking at all the different classes of antibody. But it's good to, because this is such a important time to understand the results, we can't wait to fully analyze hundreds of samples all at once. We also have to do some pilot studies so that we can, release reliable data in small bits at a time so everybody can know what we're finding. So 40 of those spike positive samples that I showed you in the previous slide were then further analyzed. So what we did is we titrated, meaning we serially diluted the milk. So, we dilute about 1 part milk and 3 parts what we call a buffer. It's really just like salt water. So we dilute, and then we keep diluting. So, you know, we get 1 in 4, 1 in 16, etc. And then we dilute the milk. It's called titrating the milk. And then we do something called determining binding endpoint titers. And this is a way, you saw that variation, right, of all those 75 samples, and the ones that mostly were positive, there's all that variation and how positive are they. So a way to get at understanding the strength of each of those responses is to determine this endpoint binding titer, which is a measurement of antibody affinity. Or it may also be measuring just literally the quantity of the antibody there. It's hard to distinguish which one it's measuring using this analysis, but it is a measure of robustness of the response. And I'm going to describe this a little bit better in a second. So when we dilute the milk, you can see each one of these COVID samples is one of those 40 that we further analyzed. And you can see here again, so this is undiluted milk, the first data point, then we dilute it, then we dilute it, dilute that, and you get this curve, which would make sense because as you dilute the milk with our buffer, you should get less and less light, right? Because there is less and less antibody as you dilute out with the salt water. And so then when we determine this endpoint titer, you can see here with this black line, what we do is we say, okay, what was the dilution of milk at a light level of 1? Okay, so you make a cutoff, and that cutoff, you then get a value of how much is the milk diluted when it hits that value of 1. And that's the endpoint binding. It's the binding endpoint, right? So you can see then you can expect each of these lines is a different sample. They're all going to have different values of how much you can dilute it while still getting that light level of 1. And obviously, if it's all the way out here, this is a stronger sample. This gray might be hard to see. So that one is one of our most potent samples because you can keep diluting it all the way to 1 in 256, right? And then you're going to still get a really nice level of light. So that's how we get a further measurement of quality of the response, the endpoint binding titer. So I'm going to show you a lot of those types of results. So of those 40 samples, which are the same ones I just showed you in the previous slide, they're laid out here, the 40 that we looked at in depth, which were you just randomly selected from the initial 75. It was found that 38 of the 40, so almost all of them, 95% of these spike positive samples also had a positive IgA endpoint titer. So meaning that at that cutoff of 1, light level of 1, they were still positive compared to the control samples, which, you know, we consider background level, right? So really not only were 88% of the screened samples positive, but then when we looked at a subset of those, almost all of them are really a high quality response, okay? And then we also do a further assessment of what we call high titer. So high titer samples, which is this dotted line here, it's a little bit arbitrary. We selected the value of greater than or equal to 5 times the cutoff value for endpoint titer, which is this segmented line here. If it goes more than 5 times higher of that cutoff, we call it high titer. So meaning you can dilute it a lot and still achieve that light level that we're looking at as the endpoint binding. And that's really for our own purposes because we're really interested in first identifying the most potent samples for further studies, like extracting this IgA and seeing if you can use it therapeutically. And then if that looks good, then we would go back and further assess the ones with less antibody, right? So it's kind of just for our own purposes, but it is interesting to see that about 50% of these samples had such a high quality antibody that the endpoint binding titer was more than 5 times the cutoff value. And you can see those because those are really the longest bars here, right? So a very robust IgA response, definitely, against the spike after infection. So then of those 40 samples, we took 20 of those, and then we also looked for other types of antibody. So first, what I'm going to show you is looking for secretory antibodies specifically. So again, this is just 20 of the samples of the 40 I just showed you, and these are the 20 that we selected. Again, it was just 20 randomly selected from the 40. And then this is the IgA data for that. So here you can see, this is again, these are 20 of the 40 samples I just showed you having that robust IgA response. And then we looked for secretory antibodies. So keep in mind that secretory antibody could be secretory IgA, or it could be secretory IgM, although there's way less IgM in milk, generally speaking. And then the way that we detect secretory antibodies, which are again the ones wrapped up in that secretory component protein that are really important for functionality in milk and other mucosal secretions. And most of the antibody in milk is in secretory form, but just because we identify IgA in the milk, that class of antibody, doesn't mean it's definitely secretory antibody. You can't know that from this analysis here I just showed you. You only know it's IgA because of the way we detect it. We can use a similar ELISA analysis using a different commercial detection antibody to say that the antibodies in the milk that are binding to the spike protein have that secretory component on them. So they are secretory antibodies. So that's a separate analysis. So in this separate analysis, you can see that, of course, these are not identical because you're not going to get identical data looking for two different classes of antibody, but you can also see that the response does look pretty similarly robust in these 20 samples where we then, again, we titrated and we determined endpoint binding titers. So 19 of the 20 undiluted milk samples from the COVID-19 recovered donors also exhibited secretory antibody binding activity. Now that's important because that's the really durable class of antibody in milk. And of those 19, 18 of those were also positive for IgA. So there's this, really enormous overlap where, essentially all samples that were IgA positive were also secretory antibody positive and vice versa. So the 18 samples, so 18 of the 19 samples that exhibited spike-specific secretory antibody activity also had positive endpoint titers. So remember, that means that the response is then very high quality. Again, just like what we saw for IgA. So lots of high quality IgA, lots of high quality secretory antibody, and then of the samples that I showed you before that we considered high titer, so really the best samples that were spike-specific with spike-specific IgA, 70% of those also were high titer for specific secretory antibodies. So what I'm getting at here is that there was a huge amount of overlap in the data for COVID-19 specific IgA and COVID-19 specific secretory antibody. So what does this mean? So this is just showing you that endpoint binding titers for secretory antibody. So what does this all mean? So what it means is that IgA and secretory antibody activity was really correlated, okay? So you can do a correlation analysis. You can look at the values that we get for undiluted milk, for IgA, and for secretory antibody, and you can basically graph them and see if there's a linear trend, right? Are they correlated? You can also take the endpoint binding titers, which is more of a measurement of quality for the secretory antibody and for the IgA, and again, you can plot those, and I don't do this on paper. There's a program mathematically that can tell you if there's a relationship between the two sets of data. No matter how you analyze it, the IgA and secretory component data is extremely correlated. So what this means is that there's an extremely high likelihood that the IgA that we're detecting is secretory IgA. And that's, again, important. Why? Because that is the most durable class. That is the functional class that is intended to be in milk, meaning that it originated probably from the gut immune response that was in response from the respiratory and the gut involvement from the COVID-19 infection. It makes sense, right? So then we get that robust secretory IgA response to COVID-19. And then really importantly for our purposes, this tells us that we should expect to be able to extract a lot of secretory IgA from donor milk if we were to go down the path of using this therapeutically for babies or for anybody. So then the last bit of this data set is that we also looked at IgG in the milk, which is still there, and it does have its importance. It is, in terms of total amount of milk antibody, present in a lower amount. But what we found is that 15 of the 20 samples were positive for spike-specific IgG as well. So again, this likely came in from the serum, from the blood, or there might have been a local production in the lymph nodes near the mammary gland. 13 of these 15 samples also had a positive endpoint titer, so they were of a good quality. What we did find is that only 2 of the samples actually were designated at high titer, and they're also just at the cutoff right there. So visually, you can see that these curves, when we dilute the milk and we look for IgG, really look different, right? So there's just way less IgG in the milk that's specific for COVID-19, and what's there is definitely just kind of a lower quality or a lower quantity in general than what we saw for IgA. Now, this isn't really that shocking because, again, IgG in general comprises way less antibody in the milk compared to IgA. So then next, to look at what we call the durability of this secretory IgA dominant response, which is what we're calling the response we see after COVID-19 infection, we wanted to see how long does this last, right? So before you saw just samples taken 4 to 6 weeks after infection. What I'm showing you now is 28 pairs of milk samples from individual COVID-19 recovered donors, and the pairs are 4 to 6 weeks and 4 to 10 months after infection. And this is just looking for spike-specific IgA, but we already know that this is really highly likely to be secretory IgA. So you can see here for the 28 pairs, this is the endpoint titer for IgA in the early time point, and this is the endpoint titer. You can follow the line. For the late time point, you can see that, you know, overall, there's not necessarily an enormous change happening. There's definitely some with more change than others, but overall, as a group, there's almost no difference between the 2 time points, okay? Meaning that the antibody overall is quite stable. It's quite durable over time. So that's what this shows. There's a 36% mean increase in endpoint titer. So really the average of all of these compared to the average of all of these, but that's not even really a significant change. You can see the averages are almost the same. 14 donors, so half of the donors had more than a 10% decrease in IgA, and close to half of the donors had more than a 10% increase in IgA, and 2 donors didn't have a significant change. So there were changes over time, but you know what's important to see here is that all the donors, all these milk donors exhibited really persistently significant positive spike IgA titers, okay? They don't disappear. And then if you take a little closer look, this is a subset of those 28. In these 14 samples, the second sample is from 7 to 10 months after infection. So these are the longest periods of time of people who've been in my study. And there's 14 people who have given me samples that have been in the study for, you know, this long, and their early time point versus late time point is 7 to 10 months apart. So that's, you know, I mean, that's quite a while, right? In terms of the pandemic, that's close to about as long as it could be at this point. And again, what you can see is that, you know, there's 5 donors with, you know, some decrease in titer. There's 8 that actually increase in titer over this time. And there's 1 with no change, and all of the IgA titers stay positive. Okay, so it's really quite persistent over time, this IgA, which is great because then you know it's still in your milk if you're breastfeeding for 10 more months. And that's just the extent we've been able to study. We don't know what happens next, but you know, it hasn't gone anywhere yet, right? And then as you keep studying and studying, you want to know what's going to be in the milk in 2 years, in 3 years even, right? What's going to be in the milk in your next pregnancy. So this is very important. And then I'm going to show you a little bit of data looking at the function of these antibodies. Okay, so again, there's different functions that antibodies have, and one of which is the blocking function, or what we call neutralization. So the way that we measure neutralizing activity is that you can either have the real virus, or you can have a virus that's harmless that has been engineered in the lab to have the spike on it. And that we actually don't use real SARS-CoV-2, although we will be doing those experiments. We use kind of a dupe virus called a pseudovirus, which is just a harmless virus that has been made to have spike protein on its surface. And it can still bind ACE2 and it still functions just the same, and antibodies can interact with that function just the same, but it won't give you COVID-19. So it's a lot safer to use. So what you do is so this is just describing that here. So this is what we use. That's called a pseudovirus that has spike as opposed to live SARS-CoV-2. So what we do is what we've done is we actually have extracted the IgA from the milk for these studies. You could probably use milk on its own in certain types of neutralization experiments, but we're not doing that yet. We're just using the extracted IgA, and there's very standard ways of getting antibody out of a fluid. So that's we do that first, and then we just mix together the extracted IgA that we dilute, just like I showed you how we dilute the milk to measure the binding activity. You dilute the extracted IgA with a constant amount of virus. You mix them together. You incubate that, and then you put that on cells that have ACE2, and you can measure how the antibody stopped the virus from infecting the cells. So we looked at 8 COVID-19 recovered samples and 8 control samples. And all of these COVID-19 samples that we used there are 8 of the ones I showed previously in all of that data, that there are 8 of those that did show positive spike-specific IgA and secretory activity. So what I'm showing you here is the neutralizing activity of the extracted milk IgA against this, what we call the pseudotyped virus that we're neutralizing. So it has the SARS-CoV-2 spike on its surface. And what you can clearly see, the solid lines are the COVID-19 samples, the segmented lines are the controls. So you can clearly see that most of the COVID-19 samples are neutralizing this virus and stopping it from infecting cells. So in fact, 6 of the 8 that we tested could neutralize the target virus. And then what's important is that the 2 that were not neutralizing actually had the lowest IgA titers that I showed you in the previous set of data. So what that means, and what we did a correlation to determine, is that the IgA binding determined by that ELISA assay and these neutralization capacities, so the function, are highly correlated. Okay, so the 2 samples that really had very low IgA, but were still considered positive, could not neutralize this virus. But the 6 that had actually high levels of antibody, as determined by the ELISA, could neutralize the virus. So that's really good news, right? And we don't know over time if that response is going to get better and better, because we know that the antibody is hanging around for a long time. And we know even that the titers in some cases are increasing over time for reasons we don't understand fully. And so that could mean that the functionality is going to change over time too. So just to summarize the infection data, most milk samples from the COVID-19 recovered participants have significant levels of SARS-CoV-2 spike-specific IgA that is highly likely to be secretory IgA. And so far from our data, the antibodies are very durable. They last as long as 10 months, and they're capable of blocking infection. And as this big study continues, we're going to fully assess the kinetics, the durability, and the function of these antibodies, and whether this response is protective for breastfed babies. And then as well, we're furthering our studies looking at if the secretory IgA can be extracted and used in a very highly targeted COVID-19 respiratory therapeutic. Because secretory IgA, as I mentioned, it's very durable, it's very resistant to degradation in mucosal tissue. And it's very unique from the IgG, which is what's in convalescent sera, which is what you've probably heard about as being used from, blood donations from people that have recovered from COVID-19. It's very different from that. And, we believe that with a good recruitment campaign, we can source so much milk from COVID-19 recovered donors, given the context we're in. And keep in mind now that we're showing that actually the antibody sticks around for a really long time. So we don't just need to target people who are recently infected. any lactating person who wants to participate in this global recruitment campaign could give milk. If they were really probably ever infected with COVID-19, they may be eligible for such a campaign. So we're actually going to be testing the efficacy of extracted secretory IgA in a small animal COVID-19 system to see, if this really pans out, and we're starting up that study soon, using, some of these various samples to use the extracted IgA to see if it can stop SARS-CoV-2 infection, in a living model. So then the very last, 3 or 4 minutes is my sneak peek at the antibody response after vaccination. So you've probably heard there have been some very recent preprints that are out about the antibody response in milk after the mRNA vaccines. And some of my data does look a lot like those, which showed, yes, there's antibodies in the milk, which is what we expected. There should be some antibody there. We know that there's, transient, there's antibodies that come in from the serum. So what we did expect that, and you'll show you my analysis gets a little bit more at an important question that we had. So what this data is showing you is that the profile overall of the antibody response to vaccination is a little bit different from what I just showed you in a lot of depth of the antibody response after infection. Now, this is not really surprising, but there has not really been a good comparison of a response between infection and vaccination, really in milk at all, because it's so understudied. So if you start on the left here, this is the IgA reactivity to spike. These are all vaccinated participants. They never had COVID-19. This is all 14 days after they were vaccinated in the solid lines. And you can see that most of them do have an IgA response to spike after vaccination. The segmented lines are the same people in the milk samples taken just before they were vaccinated. Okay, so you can see those are negative. And then after they're vaccinated, most of them generate an IgA response. Here are the endpoint titers. Again, in this case, you can already see that most of that IgA response is not surpassing what we call the high titer response. One is, the rest are kind of medium, and then there's a couple that really don't make high quality IgA at all. Then when we looked at secretory antibodies, you can see visually, and I'll show you this to compare in a second, compared to the infection data, it's not there. So in these 10 individuals who were vaccinated against COVID-19 with one of the mRNA vaccines, this is 2 weeks after the second dose, I am not seeing much secretory antibody. So there's IgA, but it doesn't look like it's secretory IgA. And then here you can see that in the endpoint titers, some of them make the cutoff of quality here, but it's not particularly robust, the ones that are positive. And then many are actually not positive in terms of the endpoint binding titer for secretory antibody. But there is a very robust IgG response, okay? So all 10 people generated a very robust IgG response, which is very different from the post-infection response. And you can see that it's variable, but most of the people, they're all considered positive, okay? These are all positive. This is the 5 times endpoint cutoff high titer line here. So you can see that most of those surpass that. And then 2 are actually really, really high. These levels are actually really much higher than what we see in infection, actually, even for the ones that are positive for IgG after infection. So the profile is very different after vaccination. There are certainly COVID-specific antibodies in the milk. They are different from what we see after infection. And then to compare in the last minute, to remind you what the profile looked like after infection, lots of really robust IgA, lots of really robust secretory antibody. Those data overlap. So this IgA is secretory IgA. Some IgG in 75% of people, but it's lower level, not particularly robust. In vaccinated participants, it's kind of flipped. There's some IgA, as there would be expected to be, because IgA is really dominant in milk, but it's not secretory. So it's probably coming in from the serum. And as I said, we do have IgA in our serum. It's in a monomeric form. It's not in secretory form. That's probably coming in from a local response from the vaccination in the lymph nodes nearby, or it's coming in from serum. What is robust is the IgG response, because that's what we're seeing in serum as well. So there's lots and lots and lots coming from the blood into the milk. And there could be some local production in the lymph nodes near the mammary gland, also increasing that in what we see in the milk. So antibodies there, it's a very different profile. And I'm going to leave you with that. So I'd like to thank everybody at my lab, especially my technician, ELISA, and the kind labs who have given us the spike protein and use of their neutralization assay. I'd like to thank everybody, some help I got at UC Merced at the Latch Lab for gifted research coordinator and materials support. Medela, of course, for their gifted support for milk shipping via Milk Stork. I'm supported by the National Institutes of Allergy and Infectious Diseases. And of course, I need to thank all of my milk donors, because I could never have this data without their participation. So thank you very much.
Thank you, Rebecca, for presenting that information for us. It was very interesting. And we do have a little bit of time, about 5 minutes, for some questions. The first question that came in is, how does a mom connect with you in order to be a part of your study?
Yeah, so you can actually, if you really just Google my name and Mount Sinai, you can find my website, or maybe that'll be passed out to everybody here, and you can find the link from my website. Also, our Gmail address is COVID-19 Human Milk Study at gmail.com. So that's specific for participating in this study. And if you email my research coordinator, we'll let you know if you're eligible to participate.
We can certainly include your contact information, Rebecca, to everyone once I send out information after the webinar. So that's perfect.
Great.
The next question that came in, Rebecca, is IgA and IgG absorbed and used by the infant in the same way?
Okay, so basically, most of the function of these antibodies is going to be in the oral nasal cavity and then in the gastrointestinal system, okay? Meaning that there's certainly an impact of, respiratory viruses, because we all know what babies look like when they breastfeed. It's bathing their oral and nasal cavities on the inside. And that's going to be, have lots of blocking activity in terms of the antibody function. And then going down into the digestive tract. In terms of, there is really no further absorption, right? Like this antibody it's called passive protection. So it's continuously being renewed every time the baby breastfeeds, but it's not a permanent type of protection. It's not going into the baby's blood system. It's not transferred like that. So the major difference between the IgA from the milk and the IgG is, if the IgA is secretory IgA, meaning it's the dimer that's wrapped up in the secretory component that's highly protective, that class will be much more long-lasting and durable to degradation in those environments. So, I don't want people to panic that after vaccination, oh my God, like it's mostly IgG, there's no secretory antibody, that meaning it's not going to be good. It's not true. So IgG still functions, but it may not last as long. And, we don't, just because, I can show you what I'm showing you, but I don't know what the real life consequence of that is going to be in the baby, right? I can tell you that IgG is still really important. That's what's in our blood. That's what does all the work. But it will get degraded faster in the baby, and it may have its most important function in the oral nasal cavity, but it may not really survive much past the stomach, whereas the secretory IgA can survive pretty well past the stomach and in the lower gastrointestinal system. So there's some differences there.
Okay, looks like we have one other question. This is just a general question that I can answer. Someone is asking if the webinar will be available as a recording at a later date. I am recording this, so we will actually be posting it on our website, and I can certainly send out an email once that's available. And it looks like we're down to almost the top of the hour. So again, Rebecca, thank you so much for presenting this information. And I wanted to let everyone know again, for the U.S. attendees, your certificates will be available on Monday by logging into your account and downloading those there. And registration is still open for the March 17th webinar. You can visit medelaeducation.com to learn more and to register for that. And the registration for the April webinars will be opening soon, so keep an eye out for that as well. And thank you everyone for joining us today, and have a great afternoon.
Thank you so much.
Thank you so much.